I wanted to read our land acknowledgement. The Eugene Natural History Society is based out of the traditional homelands of the Kalapuya peoples, most of whom are citizens of the Confederated Tribes of Grand Ronde and the Confederated Tribes of the Siletz Indians. These indigenous folks have stewarded the land for millennia. They continue to play an active role in our local communities. We are committed to supporting the many tribes and indigenous scholars and organizations working to shape the future of these lands and waters that we mutually cherish. Thank you for that. All right. So I get to welcome up board member Alicia McGraw, and Alicia is going to introduce our speaker tonight.
We take turns interviewing the speakers throughout the year, and so I was really lucky I got to have Nina, which was wonderful. So I'm going to introduce her. So I want to say it's a pleasure. I want to say it's a pleasure to introduce tonight's speaker, which is Nina Ferrari. Nina grew up in Virginia, where her two scientist parents both nurtured her early awareness and love of the natural world. Whether she was exploring nearby streams with her dad or trying to ask larger ecological questions, Nina's curiosity was encouraged by her parents. But it wasn't until she went to the University of Vermont to study wildlife biology when she took her first ornithology class and really got a love of birds. So that's where her heart settled, although she told me she loves everything. Pretty much in love with all nature. During her junior year in Vermont, Nina spent a spring semester doing field work in Chile and Patagonia, and was able to follow that up with some field work in Maine, which really cemented for her the realization that she could do this for a living, that this kind of field work was something she might be willing to pursue in the future. Today, Nina is a doctor. Oh, wait. I meant to say this. She also spent 5 years doing research at Mount St. Helens. I just wanted to say that because it didn't get in the article, but that was cool. Anyway, Nina is a doctoral researcher in the Forest Ecosystems and Society program at Oregon State University. Her work is at H.J. Andrews Experimental Forest, where she uses data gathered in the form of bioacoustic and microclimate sensors in tall trees that she has to climb many times a season. Okay, let's see here. She's attempting to use the recordings that she has and the microclimate data to create a vertical map of how birds are using the space in the up and down plane. We have lots of data on the horizontal. In recognition of her scientific leadership, field innovation, mentorship, and community engagement, Nina was honored with the 2024 Dean's Award, which is really wonderful at OSU. I got to read about you. And she also recently attended a Caltech workshop where she learned to build a neural network, basically using AI to help process her data. So she's going to talk a little bit about that today as well. Please join me in welcoming tonight's speaker, a scientist, climber, and community builder, Nina Ferrari.
Thank you so much. Just want to make sure this mic works as well. Cool. Thanks everybody so much for coming, and thanks to the Eugene Natural History Society for hosting me and treating me to a really lovely dinner before our talk here tonight. I'm going to dive in. I'm going to talk to you about what Alicia covered in that intro, understanding the vertical microclimatic gradients and bird distributions in the H.J. Andrews Experimental Forest, which I'm sure you're all familiar with. So, start off with a little bit of background, if I can get these slides moving. There we go. Yeah, so as mentioned, I grew up in Northern Virginia and was really fortunate to be exposed to field-based sciences from a really young age. I had two awesome scientists for parents, my mom, a volcanologist for the USGS, and my dad, a zoologist for the Smithsonian. I'm going to switch mics here because this one's... Okay, that seems better. They rocked and took me outside a lot and taught me about how cool nature is. And that's little me being probably a little too aggressive with a lizard. And I almost thought about wearing my hair like that tonight, but I didn't have a pink scrunchie. Anyway, Then I went to the University of Vermont for my undergraduate, where I got to take these field courses and had sort of my first foray into my own like ideas of field science. So I took some really awesome field classes, got some hands-on experience, and then after graduating, I moved out west and spent a few years working on bird-related field jobs at Mount St. Helens in southern Chile and in the Sierra Nevadas of California. And it was in those places that I started to really think about big forests and how landscapes can influence bird communities. And so from there, I got an opportunity to pursue my PhD with Dr. Matt Betts and Dr. Erica Fleischman up at OSU in Corvallis, where I study birds in forest ecosystems in the Pacific Northwest. And that's what I'm going to talk to you guys about today. I do want to first mention that my research, as I said, is conducted in the H.J. Andrews, which sits on the stolen land of the Molalla, the Confederated Tribes of the Grand Ronde, and the Confederated Tribes of the Siletz Indians. And while land acknowledgements are a necessary step, I want to encourage us all to move beyond that and towards the land back movement, which seeks to return land and sovereignty to Indigenous communities. And so with that, we'll get into my presentation. I'll go over a little bit of background. It's going to be kind of ecological theory, so stick with me during that. Go over some research questions, and then we'll dive into my 3 chapters. So I want to start by talking about how we usually think about habitat and space use of organisms on a 2D plane, but birds, which are my focus, they're not bound by gravity in the same ways that we are, and they have wings, can fly all around the forest. And this is drone footage taken by Dave Harasimchuk of a 300-foot tall old-growth Doug fir. This is one of the study trees in my research. And I want to illustrate here, there's a lot of vertical space that organisms can take advantage of. Yeah, it really just keeps going. And so there's so much like understudied space in these forests, right? We're often looking at things from a ground level, and I really thought, okay, these birds are utilizing these ecosystems in 3 dimensions, and there's a lot of unanswered questions that we can get at if we get up into those canopies. And so when thinking about how birds divvy up vertical space, we need to think about this idea of species coexistence, which is the main ecological theory behind my research. And species coexistence is the ability for organisms to coexist in the same physical space. And that's facilitated by this idea of niche partitioning, which is when species overlap in physical space without directly competing, right? They're utilizing resources, but in slightly different ways. And this phenomena occurs in a ton of different taxa, birds included. And it was first described in scientific literature in birds by Robert MacArthur in the late 50s in northeastern forests. And he found that these 5 different species of warblers occupy the same types of forests, but by utilizing different parts of the forest. So he found that these Cape May warblers forage in the tippy tops of the canopy, and these bay-breasted warblers forage in lower down branches. And I was really interested in thinking about how similar patterns play out in our Pacific Northwestern forests that are a whole lot taller than our northeastern forests. And so one factor that we know contributes to bird distributions are variations in forest structure. And so here are 2 really common forest types at the H.J. Andrews. On the left, we have second growth forest. This is about a 60-year-old stand. It's dominated entirely by Douglas fir, and you can see the trees are fairly small, and they're really evenly spaced out, and they're all about the same age. One caveat I want to make is the second growth forests at the Andrews are pretty different from your standard second growth forests in the rest of Oregon on, public or private land tree farms that are on like a 40-year age rotation. These were harvested 60 or 70 years ago and have been untouched since. So a lot of the stuff I talk about today regarding my second growth shouldn't necessarily be equated to the rest of the second growth in Oregon, because this is kind of its own little system. But on the right here, we have a picture of an old growth stand. This is about a 500-year-old stand in the Andrews. Overstory is dominated by Doug fir, but there's also a really rich midstory and understory dominated by western red cedar and western hemlock. And we see on the ground some really gnarly, coarse, woody debris that we don't always see in those second growth stands. Also, these stands are a whole lot taller than the second growth trees. And so I was really curious how these different forest structures contributed to bird distributions in these forests. Man, this thing does not want to go. There we go. Another factor that influences bird distributions is microclimate. And I feel like that's my first jargon word of the night. Microclimate, can just think of it as small climate, variations of temperature at really fine spatial scales. So at the H.J. Andrews, Sarah Fry, a previous PhD student, found that bird movement corresponds to variations in microclimate. So if we look at this plot here, on the x-axis, the lower axis, we have temperature, and on the y-axis, we have the settlement probability, so the likelihood that a bird is going to stay at a place that it settles. And we can see that for this species of hermit warbler, as the temperature increases, the settlement probability decreases. So this bird is less likely to prefer or stick around in really hot microclimates, so may be tending more to cooler climates. And so that supports this idea that microclimates contribute to bird distributions, but again, that's really just been looked at on this horizontal scale. And what I was really interested in is how do these patterns hold true when we look at them in the vertical space, given that these forests have such tall trees and complex vertical structure. So that's what I'm hoping to get at through my PhD research. And I've got 3 questions that I'm going to talk about today that guide my research. One is, how do these microclimate temperatures differ vertically in these forests? How do bird territories differ vertically in these forests? And how do birds' heights in the tree vary throughout the day and throughout the breeding season? So to get at that, I selected 14 trees across the H.J. Andrews. 7 of them are second growth and 7 of them are old growth. And I had to equip these trees with some sensors to collect my data, which was no easy feat, as you can tell by that left photo. I'm not too stoked because I had a 300-foot rope on my back that I had to carry up into the tree. But I got a lot of opportunities to climb these really tall trees, and I want to shout out Eric Forsman and Jimmy Swingle, who taught me how to tree climb, and Mark Schultz at the H.J. Andrews, who helped me wrangle a lot of these really big old growth trees. So to climb these trees, you often need to take a crossbow and shoot it over a tippy-top branch, and that has a fishing line attached to it. And from the fishing line, you can haul over a bigger piece of rope like a paracord, and then from the paracord, you can haul over an even bigger rope that you can climb on. And essentially, you use a device like this called an ascender and a bunch of other garb, and you work your way up the rope, slowly but surely. And sometimes I was climbing really tall old growth trees like on the right there, and sometimes I was zipping up some pretty mellow second growth trees like this one. Oh, that's the right. My right, your left. And eventually finding a semi-comfortable position to install my equipment, which sometimes meant doing the splits in the tree. But once you get into the canopy, there are some really spectacular views, like this one. And this, which is 82 meters, about 270 feet up in the canopy of this old growth tree. And I want us to linger here for a second. So let's think about what the environment is like at the top of these trees, right? We can see it's pretty sunny, it's pretty exposed, it's quite windy, as you can imagine, and it's pretty dry. And I want to contrast that with what's happening in the forest understory. We've got really dense vegetation, there's a lot less light hitting the forest floor, and there's a lot more moisture in these systems down here. So how do we go from these conditions at the tippy top of the canopy to these conditions in the understory? And how do those varied conditions affect birds in these forests? Well, to get at that, we first need to quantify those conditions and how they change with height. So that's my first question. How do temperatures differ vertically in these trees? And so microclimate, again, these variations in temperature at really fine scales, they've been studied very well in forest ecosystems. But most of these studies just look at microclimate in the understory, and very few have looked at how microclimate changes vertically in these forests and what that may mean for the organisms that occupy the vertical landscape of these forests. So I installed these temperature sensors, these white sensors under this PVC, at every 10 vertical meters in my trees from the understory up to the canopy. And these were recording temperature and relative humidity every 30 minutes for about 2 years. And before we dive into some results, I want to introduce this idea of microclimate buffering. So buffering occurs when temperatures in the microclimate or the small scale climate differ systematically from the ambient climate. So this is really common in forests, and you all have probably experienced it on a really hot day. You're going under some trees to seek refuge in the shade and cool off a bit. When you get a lot of trees, like you do in a forest, you get this pretty big buffering effect where temperatures in the forest are effectively decoupled from these ambient temperatures that you would experience outside of the forest. So I wanted to measure this buffering capacity. That's my visualization of buffering. And I wanted to measure how it changes as you go up the tree. And the reason I wanted to do this is, as you can imagine, this microclimate buffering can facilitate species persistence in these forests in the face of climate change, right? As the ambient temperature is getting harsher, hotter, more inclement, can birds, particularly forest birds, find refuge in these pockets of microclimate? So we're going to dive into a plot of data, and it's going to be, I think we can all get through it. So I want to show you this idea of microclimate in theory before we actually look at the data. So I'm going to show us an illustration of what this looks like. So we're comparing microclimate, so our sensor temperatures, to ambient climate. And so we're looking in our example during the summer, spring and summer. So this is relevant to breeding birds, right? So here we have an x-axis, which is the lower axis. That's going to represent our ambient temperature. And the y-axis is going to represent our microclimate temperature, so the temperature that our sensor is measuring. And we have this dashed line, and that's representing the relationship of the ambient temperature data. So that's like our plot. Oh, we got a question. I'll get there. Yeah. And then we have another line, our solid line, that's representing the data fit to our microclimate data. So in the instance that these lines are essentially over top of each other, microclimate and ambient temperatures are the exact same, right? They're getting canceled out. This is a no buffering effect. So when the ambient temperature is 8 degrees, the microclimate temperature is 8 degrees Celsius, and that's 46 Fahrenheit. I was told to make it Fahrenheit for my audience. Similarly, when the temperature is 28 Celsius or 82 Fahrenheit, in the ambient conditions, it's the same in the microclimate. So this is a no buffering scenario. We're not getting like any benefit from being in the microclimate. But let's look at the scenario in which buffering would exist. So it's the same axes. We've got our dashed line that's representing ambient temperature. And when our solid line that is representing our microclimate temperature deviates from our dashed line, we get this buffering effect. Essentially, when ambient temperatures are cooler, microclimate is warmer. And when ambient temperatures are warmer, microclimate is cooler. So we feel versed on that? Yes, broad environment. In the forest, yeah. Yeah, I'll get to that when I show my data. But the main takeaway, if you're like, what the heck was this girl talking about? If you see lines that are the same slope that are just next to each other, no buffering. If we have intersecting lines, that's microclimate buffering. That's what we want. So let's look at some actual data. So to answer your question in the front, how are we measuring ambient temperature? We're taking our tippy top sensor in the top of the tree that's essentially experiencing no forest effect. It's at the top of the tree. That's our proxy for ambient temperatures. And we're comparing every other height in the tree to that and seeing how they deviate from that tippy top sensor. So here we have same setup of the plot. We're looking at our tippy top sensor compared to our lowest down sensor. This is sensor essentially at the base of the tree. And we can see we have a pretty big buffering capacity, right? Those lines deviate a lot. The temperature is a lot cooler in the understory than it is at the tippy top of the tree. And as we work our way up the tree, now we're comparing our second highest sensor to the ambient sensor. We see that that microclimate buffering gets a little bit smaller. And here we are going a little bit higher up in the tree. It gets smaller and smaller and smaller until we get to nearly the top of the tree, and we've essentially lost all of our buffering capacity, right? So these understories of these forests are really doing a lot of work in terms of buffering against ambient climate conditions. And we see really similar patterns in our second growth trees. I just didn't include them for the sake of time. But in summary, this microclimate buffering exists in the understory and it diminishes as we go up the tree, which might have implications for canopy and midstory associated birds that hang out further up in the tree. But to answer that question, we need to figure out which species are more inclined to hang out in the canopy and the midstory, which surprisingly is not that well documented despite MacArthur's endeavors in the Northeast. So, to get at all of that, we're going to dive into my second research question, which is, do birds' territories differ vertically? So as some background, one of the main reasons that birds sing is to defend their territory against nearby competitors, right? It's usually males in the breeding season that want to stake their claim on a little chunk of land, make sure nobody else comes and takes over. And so we can use a technique called playback surveys, where we play recordings of bird songs through a speaker, and we essentially simulate the presence of a competitor. And we can document how the bird moves around the landscape as we move around with the speaker and sort of map its territory. And this is done really a lot in territory mapping, but it's almost always done, again, on the 2D scale. It's really never been looked at vertically. So I set out to do some vertical territory mapping. Oh, here's my little bird moving in response to my playback. So I use playback to draw birds to the vertical bounds of their territories, not just the horizontal. And so I have playback stations at every 15 meters in these trees. And at each station, I play a vocalization of a species that's intended to mimic a competitor that's encroaching on a bird's territory. And with the help of another person on the ground, we can keep track of where the species are responding both on the ground and in the tree as I work my way up and down doing the playback. And so we do this in both our old growth and our second growth trees. And we did this for 8 focal species that had been documented in the literature as having different height associations in the tree. So we had species that were canopy associated, mid-story, and understory. So our canopy-associated species, you all may be familiar with these. They're some of your backyard birds. We had hermit warblers and golden crown kinglets. Our mid-story species, we had western flycatcher and Hammond's flycatcher and chestnut-backed chickadee. And our understory species, we had Swainson's and hermit thrushes and Pacific wrens. So here we're going to look at the vertical heights that we observed these birds at when they were responding to our playback. First, we'll go through the old growth trees. So we had some species, like the Swainson's thrush, that really hit a ceiling at about, in this case, 15 meters up in these old growth trees. Despite me playing playback all the way up in the canopy, these birds did not want to go higher than that. We also had other species like the western flycatcher that were more associated with the mid-story. They didn't come particularly far down the tree. They didn't go particularly high in the canopy. And then we had other species like the hermit warbler that were quite shocking in that they were what I call height generalists. Instead of just exhibiting a canopy association, which is what is kind of documented in the literature, they really exhibited a territoriality throughout the whole vertical extent of the tree. And we had other species that fell all the way in between. But generally in these 3 categories, where we had understory associated species, mid-story, and a couple of height generalists. And we saw a really similar pattern in our second growth trees, where the same species, like the Swainson's thrush, were pretty restricted to the forest understory, flycatchers, mid-story, and here a little bit of the canopy. And then again, our hermit warbler being a height generalist. And what I found really interesting was despite these forests being really different in height, right? We've got our old growth trees being almost 300 feet tall, second growth trees being 100. The birds are still stratifying in these forests in the same sort of margins of the trees, despite these trees being quite different heights. So with all of that, we can start to build this understanding of what parts of the trees certain species occupy, a la MacArthur's warblers. But that was all during a territoriality context, right? Birds tend to exhibit more aggressive and intense behavior when these males are, trying to dominate their territory. So I did want to understand, how are the birds behaving when we're not probing them with a speaker? And really, how do their vertical distributions change throughout the day and throughout the breeding season? And so to get at that, we sought out to ask my third research question, which is exactly that. How do birds' heights in the trees vary throughout the day and the breeding season? Because I can't be out there all the time listening to birds and documenting where they are in the tree and climbing trees as much as I would love to do that full time. Can't do that. So I used a bioacoustics method. So remember how I installed those temperature sensors at every 10 vertical meters? I also, on those same platforms, installed these autonomous recording units, or ARUs, and those record bird songs throughout the day and throughout the season. So they're continuously recording bird songs. And using this type of data, these recordings of vocalizations, this is called bioacoustics. You'll see that word throughout the rest of the talk. And so bioacoustics data, mine resulted in, I think, 25,000 hours of data, which is way too much data to go through. If I started listening now and never slept and never ate, I would not be done for 3 years. So I can't finish my PhD by doing that. So we needed another way, which I'll get to in a sec. But it results in these beautiful spectrograms, which are essentially visualizations of sound. On the x-axis, it's time. In this case, this is a few seconds. And the y-axis is frequency. And I'm sure you all know, when birds sing, each species has a really unique song. And that also results in a really unique vocalization signature. So this, that kind of worked. This is a spectrogram for a hermit warbler. And to extract the data, we feed our recordings into a bioacoustics deep learning neural network that I built that essentially auto IDs the bird songs in the recordings. It's kind of like Merlin. I'm not going to go into it now, but Alicia mentioned that I went to this really awesome workshop at Caltech in January where I learned how to build this convolutional neural network to process all of that data. But what you need to know is you plug it in and it spits out what species are in the recording, which is amazing and helps me reduce the amount of data I have to go through. And when people use bioacoustics and they use these ARUs, they often place one in a tree in a general area, and they try to get a census of what birds are there. And that's great, but it really only tells you birds that are in the general area. But when you put multiple ARUs at known distances apart in the same tree, you can start to localize where those birds are in the tree. And so we'll walk through a general idea of how to do that. So with each of these ARUs in the tree, they're synchronized, right? So at any moment that a bird makes a noise, it's getting picked up by multiple recording units. And so that's what we're looking at here. We have 3 spectrograms. The top one is from 60 meters, the middle one is from 40, and the lower one is from 20 meters. And this is the same instance in time. And we can see, like, this vocalization here, this is a brown creeper call. So the same individual, same moment in time, captured on 3 different recordings. And you can see that this middle one happens to be the brightest, and that signifies the loudest vocalization. And the second loudest is at 21.5, and the quietest is at 61.5. And so we can do some behind the scenes triangulation, and we can approximate the height of the bird in this tree to be about here. That's where that brown creeper is. And we can do this with every other unique vocalization signature in the spectrogram. So this chestnut-backed chickadee is about here. This hermit warbler is about here. This golden crown kinglet calling is up here. And this red-breasted nuthatch is about here. And so we can do this over and over again with all of our recordings, and we can track how these birds are moving throughout the day, up and down the tree, and throughout the season. So we can do this in our old growth trees, and we can't forget about our shorter second growth trees where we can apply the same process. Sometimes we have slightly different species makeups, but same idea of mapping the vocalizations across space. So we can define how birds are moving vertically in these trees throughout the day and throughout the season, which is a big goal of mine. And then we can link these data back to the data like our vertical microclimate data from the beginning and vertical vegetation structure data. So remember our old growth and our second growth trees, they're really different. They vary in height, they vary in canopy cover, they vary in vertical vegetation structure. And we can use LiDAR, which is essentially a laser shot down from a drone. And every surface that it hits, every leaf, every branch, it reflects back up and it kind of logs that as a data point or a point cloud. And we can get these really fine detailed vertical maps of our trees. These are actually two of my trees for my research. And this is the quantity of data we can get from LiDAR flights, which is really cool. And so this gives us really, really fine resolution, vertical patterns. And so my hope is that we can tie in this bird movement data with the microclimate data and the vertical vegetation structure data to try to paint this picture of what is it that's driving this vertical movement of these birds. Are they, going down into the understory on these really hot days because there's microclimate refugia there? Are they hunkering down in a middle canopy because that's where there's a lot of foliage and perhaps a lot of prey? That's the answer I'm just ultimately trying to get at with these data. And so I want to highlight a few takeaways from my research that is still very much in progress. But we can see these microclimate buffering patterns particularly strong in the understory and particularly strong in the old growth forests. And there is this, there are different species that occupy different vertical territories in these trees. And we can use bioacoustics to understand how these vertical associations change throughout the day and throughout the season. And we can relate those bird movement data back to our microclimate and LiDAR data to get a better picture of how birds are utilizing these forest ecosystems. And before I wrap up, I want to make a note that all of these data were collected before the Lookout Fire. I'm sure a lot of you know. In 2023, in August, the Lookout Fire, lightning fire, ignited at the Andrews and burned about 70%. Unfortunately, about half my trees were burned and killed, including this 500-year-old tree we called Wolfie. Wolfie is a common word used to describe these really big, gnarly old growth trees that have these wild epicormic branches and incredible vertical structure. And on your left is a photo of me ascending into Wolfie to collect some data in 2023, just a few months before the fire. And then this photo is from May of 2024, the first time I was able to get back in. And you can see that a lot of this vertical structure obviously has changed in the fire, right? This tree died, it lost all of its foliage, it lost some branches and some small trees around it. And while I don't have the capacity to look into this stuff in my PhD, it is really important for people to think about how these big disturbances like fires impact these forests from a microclimate refugia perspective for these organisms like birds and other vertically associated organisms like salamanders and tree voles. We often think of fire and we look at fire maps on this, again, this 2D scale, and it can be catastrophic feeling, but we also need to think about how it's affecting these 3D habitats. So I revisited Wolfie in early May, and as I approached, I found the big steel link and the pulley that I used to climb this tree. So this used to be at the tippy top of the tree, and it must have fallen when the fire got to the canopy and melted the nylon webbing that was holding it up. But I found that at the base of the tree, which was cool because I got to take a gift home from Wolfie. But I did start to get a little sad, a lot sad, thinking about how these trees that I got to know and got to learn so much about myself climbing, how could these hundreds of year old trees that I was here for what I thought was a blip of their lifetime, how was it that I witnessed their death? And I got really sad thinking about how other people aren't going to get to experience these trees in the way I did. But as I was hiking out of that stand, I saw this big stump of an old growth tree that had probably been cut a long time ago, maybe probably the late 50s when the Andrews was originally harvested. And it's a huge stump. It's way bigger than, it's probably about the same size as Wolfie. And in it was this hemlock, which you can see growing up. You can see the trunk of. And that hemlock still had needles on it after the fire. So even in these big trees' death and these catastrophic disturbances, we can think of these, oh my gosh, this tree died. It's no longer providing this habitat for these birds, like how devastating. But these trees continue to provide life in their death. These old growth trees are going to remain standing and they're going to slowly decay, and that's going to provide habitat for birds like woodpeckers and other cavity nesters like owls. And it's going to slowly break down and provide nutrients into the soil. And this tree is going to live on despite it feeling like this drastic end of life event. So I wanted to end on not too depressing of a note. So perhaps Wolfie will one day, be a nice little seed bank for a hemlock to grow really tall too. And yeah, I couldn't have done all of this research without a lot of people. I want to thank my advisors, Matt Betts and Erica Fleischman, my committee members, a lot of mentors at OSU, and some really incredible field crews that helped me get out there every summer, and a lot of funders that paid the bills. And I want to leave us with another video. So this is a video of one of my trees that actually survived the fire. This is a pre-fire video, of course. But this is a canopy of an old growth Doug fir. And I want to give you guys a little bit of a treat of what it's like to be up there on a beautiful sunny day. And with that, I think I have plenty of time for questions. So thank you.
Wow. So you know the drill. You ask the question, I repeat the question, Nina answers so that our Zoom audience can hear it. Greg.
Yeah, fascinating. So I wondered what resources there are for birds that predicate this zonation. They're all eating insects, presumably, but many of them are also nesting. And so the resource for them, I guess, is the moss and the lichens and branches in the tree. Can we assume that the ground nesting, the ones in the lower part are all living on the ground? What are the resources that are implicated in this?
So let's see, I'm going to try this. So Greg's question is, there are resources that apparently the birds are partitioning that could be prey or even nesting material and that sort of thing. And I think you finished with, what are the resources that birds on the ground might be partitioning, or all of them for that matter, at any height?
Yeah, that's a good question. Main answer, we don't really know. It's hard to know. When I was originally dreaming up this study, I was also going to try to survey arthropods vertically in trees, but that's apparently harder than any of this. But so we have inclinations, right? There's been some diet analyses of some of these birds, including at the Andrews by some undergrads, which are really awesome. Likely, mostly the canopy birds are eating things like flies and spiders. Those are the big, and lepidopteran larvae like caterpillars. Understory species like the thrushes, they're, the ones that kick and scrape and then try to eat little arthropods in the ground, sometimes fungi. But I think ultimately we don't really know how they're partitioning and what they're partitioning, right? Are they partitioning temperature? Likely not. Likely they're not reaching their thermal maxima and they're like, oh, I'm so hot, I need to go downstairs or whatever. Probably temperature and vegetation structure are proxies for other things like prey availability, but I don't have a solid answer for you. That would be a future PhD student's question.
Did MacArthur find anything?
Yeah, he did actually. Oh, go ahead.
Yeah, so his follow-on question was, did MacArthur find anything that they were partitioning?
So he did find that they were okay, so I should caveat MacArthur. Awesome research. but he did do it all from the ground. So he was doing these visual surveys, and he was mostly seeing that these birds are foraging on different parts of the canopy and in different like fashions, but he didn't actually there's not a lot of data on what they were actually eating. Likely, again, those arthropods distributed in the canopy. But another important point about those northeastern forests is they have a lot more broadleaf deciduous tree diversity than we do out here, and that tends to be more palatable for herbivorous arthropods like Lepidopteran larvae. So there is a richer suite of food resources in deciduous forests for birds than there are out here. So probably foraging on slightly different things than our warblers.
John, you already got one.
Thanks.
Curious, like as temperatures increase because of climate change, how you see some of the microclimate bufferings changing as well, and is impacting birds' habitats, like if they're having to shift and move around.
Right. So the question is, how is climate change also changing the buffering regime, and how might the birds respond to that?
Yeah, that's an awesome question. So one thing is we don't really know because we're not feeling the extremes that we likely will. But if you remember that figure I showed you with the two lines deviating, the further to the right we got, the further those lines deviated. So the idea is if we follow those linear projections, that understories would continue to buffer as temperatures get hotter, and that buffering capacity would increase. There's another potential theory that would suggest that like these systems are completely decoupling. Other microclimatologists posit that perhaps they're not going to totally break away and experience completely different patterns, but perhaps they'll still be muted to some degree. The like catastrophic idea is that, you know, at some point we'll reach a tipping point and they won't serve much purpose at all. But generally what we see in our data is even on these really hot days, we still have cool understories. So I don't want to speculate what that means for birds per se. I would imagine that understory birds might be more comfortable, but they also might be facing increased competition as other birds shift down in the tree. That's hopefully what we'll figure out once we, you know, weave all my data together. So I'll have to get back to you.
John's up next.
Species that kind of specialize for towards the top. That lower and all every place, but nothing.
Yeah.
So the question was, why are there no species that just specialize in the top?
Yeah. Well, I only looked at 8. And that's because those are 8 that are really commonly distributed. So we could do them in all of our trees. Hermit warblers are a tricky bird because they're often thought of as this canopy associated species, right? A crane your neck warbler that you're, you know, looking through way high up in the tree, but they're also a really aggressive warbler. So they are very responsive to playback, and they will shoot down from the canopy and come, you know, essentially to your speaker. And so they may be like perhaps foraging more in the canopy, but when they feel a threat to their territory, they're a little bit more activated than perhaps some other species. But there may be other warblers like the black-throated gray warbler, which is a congener of, so same genus, as the hermit warbler, that we unfortunately don't have a lot of data for because they're more of a broadleaf forest obligate, but maybe they may be more canopy associated. I suspect that like if we were to expand the study to more species, we would see some more stratification.
Tom.
Mm-hmm.
So the question is, can you really use vocalization as a proxy for presence, or might there be some silent birds that are getting by unnoticed?
Yeah, well, great question. And I feel like you must have been talking to my committee members because that was something they would have asked me. I would argue, you know, we always survey birds through vocalizations. You know, that's the standard way. We go out in the field and we listen with our ears and we write down what we hear. Rarely in these forests and really tall trees do we see all of the species. But yes, birds exhibit vocalization patterns, particularly during the breeding season, males sing in the morning, and that is the time that you go out and survey birds. And that is what gets a little bit difficult with this bioacoustics data is as you get later in the day and later in the season, after these birds have nested, their vocalizations decrease. And that is a big question that we're always trying to get at in avian ecology is how do we track these birds without necessarily using GPSs, without necessarily with knowing that they're not going to vocalize forever. They're going to vocalize at this specific time. And that is not to get too into the weeds of that classifier I built, but the classifier can recognize like songs and calls. So the hope is that while yes, males mostly sing in the morning during the breeding season, a lot of birds call at all times of day. And so if this classifier can pick up on calls, we can track where birds are calling and singing. But it's certainly a huge limitation of bird studies is without following the birds around all day, we're using their vocalization as a proxy for their presence. It's been held up in science so far with the assumption that if they're singing, it's a male that has a territory. If it's singing continuously, it's probably there.
So we're going to take a little break and give a shout out to all the people that are joining us online. Thank you. I will say, however, the cookies are really especially good tonight. And Alicia has a couple of questions showing up on the chat.
Yeah, all of them except two, the chestnut-backed chickadee and the Pacific wren. Those are residents, but the rest migrate. Mm-hmm. Do you know what the balance
appears to be about 12 degrees Celsius? The person says, which seemed constant at varying heights. Do you have any idea why the balance point is 12 degrees and not 6 or 20? Is there some relationship possibly with evapotranspiration, something that's going on? They're trying to figure it out. We got a great audience.
Yeah, that's a good question.
So I think I have to direct the Zoom audience to the chat for that one. I'm not even going to try.
I can try to summarize it. Essentially, they noticed that the inflection point of those microclimate figures happened to be at 12 degrees Celsius, and that was consistent across heights. I will say that the data that we were looking at was just summer. And so spring and summer. So when you look at the whole year, that inflection point slips down. And that figure looks more balanced, right? It was really like lopsided. There was a lot of buffering against warm temperatures, and it looked like there wasn't a lot of buffering against cool temperatures. I want to dispel that. If you look in the winter, there is also buffering against cool temperatures. So microclimates are warmer in the understory than the rest of the tree. But that's okay. So, oh man, I could go on. There's a lot of really cool.
You have time.
Okay, great. Yeah. Kick me out. There's a ton of really different features that influence microclimate, and they occur at different scales. So if we think about like the big scale, the planetary scale, the way that the Earth is tilted, right? What season we're in influences how much solar radiation we get, right? The sun is the big driver of our microclimate here, our temperatures. So depending on what season you're in and what part of the world you're in and when you're in that part of the world, there's also like broader landscape level scales like elevation and topography that will influence microclimate, right? You have this thing called adiabatic lapse rate, which is as you move away from the Earth's surface, so increase in elevation, you get cooler. That's hardcore physics, not quite where I'm at, but and then you also have these like ridges and valleys that can contribute to cool air pooling and surface level temperature differences. So you have this big planetary scale stuff, you've got this landscape scale stuff, you've got this forest scale stuff, right? You've got this vertical vegetation that's interfering with the solar radiation. So that's like kind of in the canopy cover that's sort of blocking all of the solar radiation. But you also have the tree scale where trees are actually playing a role in buffering in that their evapotranspiration is releasing moisture and gases into the immediate environment, and that is interplaying with microclimate. So it is really hard to know like why that balance point, where and when and why that balance point hits. There's like a ton of things. that could be contributing to why those are balancing consistently. But it's probably, if it's consistent across all of our trees, we could posit it's a bigger like landscape feature. Perhaps if we see variations in that across our trees, maybe it's more of like a stand level feature. And if we see variation of that in height, perhaps it is like a really localized feature. That's an awesome question. Sure, yeah.
Of course. Here, I'm going to give you the mic, though.
Also, before we do that, Nina, you need to tell these people how many times you climbed those trees.
Oh, I don't know, but
Many times.
like a lot of days. But I think I did the math once, and it's something like, I think I've climbed 22,000 vertical feet. Yeah.
I read an article about you, and it said that you had climbed every tree in the first season 50 times.
Yeah, that
So
probably
that's
is right.
just to give people. I just think they should.
Yeah,
You should
that's
get
great.
some credit for that. Okay, so here's Dave's question. It says, "Do you think the presence of nitrogenous lichens on the forest floor of old growth, that are much fewer in the second growth, might represent special resources for the nutrition cycle?" But I think he means nutrient cycle.
Yeah, I have no idea. That is out of my wheelhouse. I'm sure there's some lichenologists in the audience that could take a better stab at that one. Yeah, I mean, it sounds like there are more lichens in old growth and that they contribute to this nutrient cycling from what his question sounds like. So perhaps that's contributing to like richer arthropod abundance or some other abundance that would confer benefits for understory associated species. But who knows? I wonder, there's a lot of lichens in the canopy too. So I wonder what they're doing up there.
Connie.
Done by you, but by others, and so I don't know how you were able to integrate if there were specific species studies, longitudinal species studies of these species, that you were able to integrate into your type of research to get a better idea of the horizontal spread around the area as well.
So
Yeah.
I think the question was, how were you able to integrate all of the previous data that existed probably for a lot of these species into your own work?
Yeah, so there is a wealth of data, particularly for these species. There's a big love for old growth in the world. But a lot of that is on the horizontal landscape. There was one study, or two studies actually done by Dave Shaw at OSU using a canopy crane at the Wind River Research Station in the late 90s, early 2000s, where he was documenting where birds were in the vertical strata. And his research very much inspired mine. But other than him, there's not been a lot done in the Northwest of where these birds are. There's not been a lot of vertical work with birds, and the bulk of it's been done in the tropics, where there's a lot of diversity.
Yeah, in the back, almost the back. You don't have to look back. You can just ask your question. Right there. Sorry, I don't know your name.
There's some differences between the growth and the buffering capacity. Was there any other differences between the second growth and old growth, such as the distribution of, or the population of birds that visited the second growth versus the old growth? Do they prefer one over the other? Related to the microclimate or not?
So the question is, were there differences between the old growth and the second growth in terms of maybe even not just the number of birds, but the kinds of birds that populated one of the other sorts of stands?
Yeah. So I guess one caveat I want to make is there's like not a lot of temperature difference between the trees, the second growth and the old growth. What we do see is there's more space in the old growth, right? There's more vertical space in these taller trees that serve as buffers. So I just wanted to make sure I clarified that. So this is actually a perfect visual for this. Okay. Here in the forefront of these shorter, brighter green trees, these are second growth trees. And then right just above it are taller old growth trees. And this is very much how the landscape of the Andrews looks. It's like this matrix patchwork of second growth and old growth. And so because of that, we actually, well, I don't know if it's necessarily because of that, but coincidentally, we see a lot of the same species of birds in these forests, right? They're the same tree species. They're really similar forests. They really just differ in age and height. And so I would say we get a few more old growth obligate species in the old growth trees, but for the most part, the species distributions are really similar. But I think the date, once we pump through all that bioacoustics data, I'll have a more solid answer for you.
Okay, I'm going to take one more. You got it.
I guess sort of how to get to existential.
Let's get there.
That's the last question.
Yeah.
What do you see as the implications or future applications for your study? Like what do you hope this data will bring to the
So the question is, what are the future for these data in terms of implications and get it? So that's an open-ended one for you to run with. At the
Sure.
end.
I appreciate that question a lot because I think a lot of the times we scientists like to talk about the science and sometimes we forget about the application of the science. So the hope for this research is that one, it's going to illuminate the importance of old growth for microclimate buffering, right? We now more than ever, unfortunately, have old growth that is at risk of being logged and its protection being lost. So I'm hoping that this is another feather in the quiver of our argument for old growth. But what does that look like? Okay, we can protect all the old growth in the world and it still might not be enough habitat. But can we manage some of these second growth trees and second growth stands to have more old growth structure, have more characteristics that will confer this microclimate buffering? Can we manage for taller trees? Can we manage for more vertically complex trees that we know will kind of create this hospitable refuge for birds? That would be my hope.
Great answer. I got to ask my question. Oh, what, you're going to force one more on me? Okay.
Woo, go science. Am I right?
So that was a yay science. I got to ask my question at dinner and I promised I wouldn't be disappointed, but I really kind of was. Were there any salamanders up there?
Sadly, not that I saw.
Thanks again to the Zoom audience for coming in, and let's all give Nina another big hand for a great talk.
Thank you.